Hello students, welcome to today's class on biochemical test in clinical microbiology. In this session, we will learn the various biochemical tests used for the identification of the bacteria in the microbiology lab. So what are the learning objectives? First, understand the principle, know the procedure and interpretation, differentiate important bacteria using this test, apply this test in the diagnosis of infectious diseases. What are the key points? All the biochemical tests are in vitro tests mainly performed in the microbiology lab. Bacteria produces the enzymes and these enzymes help in the biochemical reactions. I will discuss all the biochemical tests one by one. So let us start with the first important biochemical test, that is catalase test. So what is the purpose? To differentiate members of the micrococci from the members of the streptococci. So look at the chemical reaction. Catalase enzyme, which is mainly present in the bacteria, decomposes hydrogen peroxide to water and oxygen, and this oxygen is mainly responsible for the bubble formation. Catalase enzyme mainly present in the aerobic and facultative organism and absent in the anaerobic organism. And hydrogen peroxide, if allowed to accumulate in the bacterial cell, it is lethal. So catalase enzyme is mainly helpful for the bacteria to survive inside the human body. So what are the requirement for the catalase test? First, clean glass slide, wooden stick, nutrient agar plate, stick to the microorganism, 3% hydrogen peroxide. Then what is the procedure? Take a clean glass slide, transfer the colony of the microorganism from the nutrient agar plate to the surface of the glass slide, then add 1 drop of 3% hydrogen peroxide and observe for the bubble formation. Then what is the interpretation? Rapid and sustained appearance of the bubbles indicates positive test. On the right side, the test is positive, and on the left side, absence of the bubble indicates the test is negative. Applied aspect of the catalase test in the diagnosis of the infectious agents. Catalase test positive means the organism is most likely Staphylococcus species, and catalase negative means Streptococcus species. Clinical application, suppose a person showing non-positive cocci in clusters is tested with the catalase. Catalase positive likely indicates staphylococci and negative likely streptococci or enterococci. Catalase positive, gram-positive bacilli are Listeria monocytogenes, Corynebacterium, Bacillus. Then all the Enterobacteriaceae family are catalase positive except Shigella dysentery type 1. The other gram-negative bacilli which mainly show catalase positive are Vibrio cholerae and Pseudomonas species. Catalase negative organisms are Clostridium and Erysipelothrips. So some important points during catalase test. The first point is do not use blood agar plate. Why? Catalase present in the red blood cell. If we use blood agar plate, there may be chance of carryover of catalase which leads to false positive result. Second, do not use inoculating loop because nichrome metal present in the inoculating loop interfere with the biochemical reaction which give false positive results. Thank you. Kindly like, share and subscribe to channels for getting more updates.